What's up, boys and girls and everyone else? This is another science video lesson episode right here. And for this episode, we will be going to talk about the different effects in both genes and chromosomes if there are changes happening in the DNA. Come join me today as we go over the different genetic disorders and the concept of mutation. Alright, so here we are in our lesson for this episode. Okay, so but before we go to the main lesson that we have right here, so let's take a look at this picture. Okay, so this picture has been circulating the social media for several times, and yes, I somehow agree with this picture that you have right here. The mutations in real life is somehow just like the gentle dog on the picture, and the mutations that you can the perspective of mutation that is seen in the science fiction movies are somehow the way it is perceived in this picture. Okay, so you might want to say that you just hope that the mutations were just like in the science fiction movies, but fortunately enough, it's not. All right, it is not. So mutations in real life, it's somehow neither good or bad at the same time. They are somehow there are some mutations that are neutral. Okay, so that is what we will going to talk about in this particular lesson that we have right here. All right, so I think you just you are familiar with this movie. You are familiar with this movie. I think you are familiar with this. So this is one of the comics or the movie adaptation of what they call X-Men. All right, so X-Men are a group of mutants. Alright, mutant individuals that has a certain mutation in their body, and this mutation produces certain abilities that a normal person can't do. Alright, for example, we have here Professor X, which can control the mind of the people, and he can read also the mind of the people. Alright, so next one we have here Mystique. Alright, so we have here Mystique, so he can or she can change herself into different forms. It might be another person, it might be a male or a female, something like that. So we have here Quicksilver, something like that. Okay, so Quicksilver, his mutation enables him to run fast. Alright, and we have here Angel. Alright, so we have here Angel over here. I don't know what, I forgot his name. Alright, so now his abilities, his mutation enables him to fly, and he has a steel wings that you have right here. Alright, so we have here Storm. Alright, so her mutation gave her the ability to cast lightning or even control the wind. Alright, or enable to produce storm or something like a hurricane, something like that. Alright, so Magneto. Alright, so who can forget Magneto? So Magneto is somehow a mutant, and his mutation gave him the ability to control metallic objects. Alright, so if somehow in real life, if somehow these mutations are real, so might likely that some people in this planet will be able to gain this particular ability that we have right here. But unfortunately, it's not. All right, the mutation that is the mutation that is or that was seen in this movie is different from the real mutation in real life. All right, so that is what we were going to discover today: the mutation and its genetic disorder. All right, so the genetic disorder that it is associated. Okay, so let's get right into it. Alright, so what is mutation? So mutation is a change in the DNA sequence. Alright, so in the previous video that we have right here, in the previous video lesson, we talked about how the DNA replicates and how the DNA creates protein. Alright, via protein synthesis. Okay, this central dogma. Okay, so this is mutation. Mutation happens when there is changes or there are changes in the DNA sequence that may affect the whole chromosome or just one part of the gene segment of that particular DNA. Alright? So, that is mutation. Now, there are two kinds of mutation. Okay? According to where it happens. Okay? So, the first one, we have the chromosomal mutation. And the second one is the gene mutation. Alright? So, as you can see here, these two mutations that you have right here occurs in two different places. Okay? It occurs in two different places. The first one is in the chromosome. Alright? It affects the number of the chromosome or the structure of the chromosome. Or the second one, it is on the gene itself. Okay? It is on the segment of the DNA. So remember that genes are segments of DNA. So if there are changes in these segments of your DNA, then therefore mutation has just occurred in that particular segment. Alright, so these are the two things that we will going to discover this in this video lesson. And what are the definite causes of mutation? So there are several causes of mutation. Alright, so the first one is the mistakes in the DNA replication. Alright, so most of the mutations are caused by mistakes in the DNA replication. Alright, so as we recall that particular lesson, alright, DNA replication is the process by which the DNA make itself a copy. Okay, make itself an exact copy. And DNA replication is very important in terms of growth and maintenance of body parts. Okay, so if the DNA fails to do that, then therefore mutation can occur. The second one is external influences. Alright, so we have here chemicals, so in the form of what they call carcinogens. Alright, so carcinogens. So carcinogens are found in some food that contains these chemicals that can cause or that can trigger mutation. Alright, next one is radiation. Okay, so if you recall our physics, okay, our physics lesson about electromagnetic waves. Okay, so the link is above, right? Okay, the link is above, is on above. Alright, so you can click that link in order for you to have an idea what is this, what are these radiation that can cause certain mutation in our body. So we call these radiations as ionizing radiation. Alright, so ionizing radiation. Okay, so these are a type of electromagnetic radiation that has the capability of knocking out the electrons from its orbit. Okay, now if the electrons were knocked from its orbit, it will definitely change the structure of that particular molecule, and once it changes its structure, it will affect its function as well. Okay, so radiation. All right, next one is infectious agent. For example, viruses. Okay, bacteria, okay, protozoans, okay, something like that. Okay, so those are some infectious agents, okay, that can cause mutation of a certain segment of the DNA or even the chromosome. All right, so now these are the chromosomes. These are okay, these are the parts of your nucleus. Okay, they are located in the nucleus, and this is where the DNA is located. Okay. This is where the DNA is located, and these are made up of DNA. So, something like that. Okay, so chromosomes contains this genetic information that we have in our body, and once the chromosomes are affected by these changes in the DNA segment, it will definitely changes what form of an organism are we talking about, and how this particular body part functions. Okay, it will affect a lot. Okay, so in order to start our discussion with what is mutation is all about, okay, let's start first with the gene mutation. Okay, so the gene mutation is a type of mutation in which the DNA sequence is altered. Okay, so this is a type of mutation in which the DNA sequence of a certain cell has been altered. Okay, once it is altered, it will create a lot of effects. Okay, it will create a lot of effects on the functionality of that particular cell. Alright, so we have here the first the first classification of gene mutation that we have right here is called the hereditary mutations. Alright, so from the word itself, heredity, it means it can be inherited from the parent and it can be passed through the offspring. Alright, so these mutations are said to be germline mutation because these mutations are present in parent's egg or even sperm cell. Alright, so these kinds of mutation or the hereditary mutations are present in parent's egg or sperm cell, or definitely in the gametes of the parent. Okay, so that is a hereditary mutation. Okay, so the next classification of gene mutation is the acquired or the somatic mutation. Alright, so the acquired or somatic mutation. Now this, from the word itself, acquired, it means it is caused by external means. Alright, so this is caused by external, so or some environmental factors cause this particular mutation that we have right here, the acquired or somatic mutation. Okay, so these changes are caused by somewhat like radiation, okay, ultraviolet radiation, or exposure to X-ray, exposure to radioactive material, something like that. And because of these particular environmental factors, it can lead to mistakes in the DNA replication. Once there is a mistakes in the DNA replication, it will be carried out. Okay, it will be carried out through cell division like mitosis or even meiosis, and it will affect what kind of offspring you will produce, or what kind of body part, or what will be the effect on the body parts. Okay, it will likely to have. Okay, example, the functionality of a certain body part will be affected or something like that. Okay, so that is the acquired or the somatic mutation. Okay, so the next part of this lesson that we have right here, we will going to find out what are the different gene mutations, okay, that we have right here. Okay, so let's start now. Alright, so we are here in the first example of gene mutation, and we have right here the deletion. Alright, so deletion is from the word itself, delete, which means that in a single nucleotide sequence, so as you can see right here, so on the top portion, this is the original or the normal DNA sequence. Okay, and the normal DNA sequence is written as in codons. We have here the CAT, so on and so forth. Alright, and the CAT codon, or the CAT codon that you have right here, translates into histidine amino acid. Alright, the one that you see here. So all of the sequence that you have right here on the DNA sequence that you have right here is translated as amino acid called histidine. Now, in the deletion process, there is a single nucleotide on that sequence. There will be a single nucleotide on that sequence that will be deleted. For example, if the sequence is written as, or if the sequence is arranged as CAT, and eventually the letter A has been deleted, okay, has been deleted on the sequence, what will happen here is it will affect how the amino acid, or what kind of amino acid will be translated. So as you can see, removing the A from the sequence affects what kind of protein will be produced in the end. Okay, so instead of histidine, because of the removal of the nucleotide adenine in the sequence, what is produced now is leucine, because instead of having CAT, we have now CTC. Okay, we have now CTC, and instead of having CAT, we have now ATC. All right, on the subsequent on the next sequence that we have right here. All right, so instead of having histidine, we have now isoleucine. All right, now this particular amino acid can cause malfunctioning of a certain body parts or a certain protein. Okay, it can be a hormone or an enzyme that is used in other functions. Okay, so if this happens, that particular body part, if that is a body part, that particular hormone, okay, that particular hormone or as an enzyme that corresponds to this particular protein, when it undergoes, or when the sequence of the DNA that used to create that particular protein was deleted, what will happen here is it will render, or it will produce a malfunctioned protein. Okay, so some examples of diseases that is caused by deletion is the deletion of number 5 chromosome, the one that you have right here. Okay, so the deletion of number 5 chromosome that you have over here is called the cat's cry syndrome. Alright, so the cat's cry syndrome. So, cat's cry syndrome. Okay, so the cat's cry syndrome, okay, is characterized by an individual that has a smaller head, okay, unusual facial features that you have right here, which is different from a normal person, and most important characteristic, or most noticeable characteristics of people that has cat's cry syndrome is high-toned cry. Okay, high-pitched cry, so which you will hear like this one. Okay, so that is a cat's cry, that is a high-pitched cry of a person that has this kind of syndrome that we have right here. Okay, most of the individual that has this kind of syndrome, again, it has a unusual facial features, okay, smaller face, okay, it has a smaller face, and some of them has intellectual disabilities, all right, and sometimes they have this kind of characteristics that they have trouble breathing, okay, so, and many more. Okay, so this is the cat's cry syndrome. Okay, so this is the cat's cry syndrome, and one of the causes of the cat's cry syndrome is the deletion of the chromosome, or parts of the chromosome number 5. Another disease which is caused by deletion, okay, deletion mutation, is the cystic fibrosis. All right, so the cystic fibrosis is a deletion type mutation in which a certain protein, okay, which is used to make, or which is used to secrete a normal amount of mucus in the lungs and in the digestive system, happens to be malfunctioned. Okay, it happens to be malfunctioned. So what happens here? The lungs and the digestive system, or the gut, okay, secretes a lot of mucus which is unnecessary. All right, it secretes unnecessary amount of mucus and blocks this particular organs, like the lungs that you have right here. So this mucus blocks the lungs, and the person who has this kind of disease has difficulty in breathing. Okay, so it has the person who has this mucus inside the lungs is usually vulnerable to respiratory diseases. Okay, all right, and also these people also, if it affects the digestive system, it blocks the pancreas. So when you block the pancreas, you will have a trouble, or you will have something like an effect in your metabolism. Okay, so that's the reason why this person that you have right here has certain has a skinny, okay, has a skinny body feature, which is an effect of your disease, right, like the one that you have right here, which is called the cystic fibrosis. All right, so these are the two diseases caused by the deletion of a certain sequence of the DNA, which is called the deletion process. Okay, so So the next type of gene mutation, or the next kind of or example of gene mutation that we have right here, is called the insertion mutation. Alright, so compared to the deletion mutation, so this is again the original. Alright, so this is again the original. So compared to the deletion, so what happens here is, out of nowhere, okay, out of nowhere, or it might be caused of chemicals, radiation, or even caused by viruses, something like that. There is a nucleotide that was inserted to your sequence. Alright, so there is a nucleotide like the one that you have right here. In this example, we have here the adenine. Alright, the adenine was inserted forcefully in your sequence. So, what will happen? So, logically speaking, it will affect the whole sequence of your DNA that is shown right here. So, once it is affected, again, the normal translation for that particular protein will also be affected. So, what happens here is, instead of producing histidine, alright, assuming that it is the normal amino acid that was produced in the normal sequence of the DNA, what happens, or it happens to be that the one that is produced is rheonine. Alright, so the rheonine was produced because of the forceful entry of the adenine that you have on your sequence. Now, it will also affect the next or the consecutive sequences that you have right here. Instead of CAT, it becomes now the TCA. Alright, it becomes, the codons become now TCA. So, instead of histidine, it becomes now serine. Alright, now, if this is a protein that was coded to perform several functions or normal function, so what happens is, if there is a mutation like insertion on that particular sequence, it will affect that particular protein. Again, you will produce a malfunctioned protein. Okay, so this is the insertion mutation. Okay, so let's find out what is the disease. Okay, let's have an example of a disease caused by insertion. So we have here the Huntington's disease. Okay, so the Huntington's disease is a disease in which the nerve cells automatically degenerates. Okay, because of the malfunction protein brought about by the insertion process, there is now a degenerative process that is happening in the brain cell of this particular person in the photo. Okay? So, the nerve cell, instead of development, or instead the nerve cell develops into a more robust and more functional nerve cell, it degenerates. Okay, once it degenerates, what happens to your central nervous system will affect your movement. And not only that, it will affect your intellectual abilities. And also, it will affect your emotion or even your mood or something like that. Okay, so this is the Huntington's disease. These are the effects of the Huntington's disease. Okay, and again, Huntington's disease is a product, or it is the cause, or it is the disease caused by insertion. Alright, so let's go now to the next example of mutation that we have right here, gene mutation, which is called the inversion. Alright, so inversion, unlike insertion and deletion, alright, in inversion, what happens to a part of the chromosome or the segment of the DNA is, this is the normal segment. Okay, this is the normal chromosome or the normal segment of the DNA. Happens to be, it is reinserted backwards. Okay, if this is the normal, and during the inversion mutation, what happens is, the segment of that particular chromosome that you have right here was inserted interchangeably. Okay, so do you think that is normal? Okay, do you think that is normal? No. Okay, so what happens here is, if this particular normal chromosome secretes, or rather creates, an amino acid. For example, let's assume that this normal set of chromosome creates an amino acid called serine. Okay, for example. if that particular normal set of chromosome creates serine, now if you have an inversion, it will definitely change what kind of amino acid it will produce. So assuming that the inversion happens, it will now create an amino acid which is completely different from normal. So it will become like, for example, threonine. Okay? So, which is, if that is a protein necessarily, or if serine is a protein necessary for your body to function properly, then therefore, changing that amino acid will definitely affect that particular protein. So in short, at the end of the day, the protein that will be produced after the inversion process will be a malfunctioned one. Alright, so let's take a look. Okay, this is the southern blot. Okay, this is the southern blot, and what you see right here are lanes of DNA. Alright, so segments, these are segments of DNA. As you can see from number 2 and number 6, they are normal. This is what the normal DNA sequence of a person that has a normal blood clot. Okay? A person that has normal blood clotting. Okay. Now, we have here lane number 1. This is a male that has a hemophilia. So what is a hemophilia? Hemophilia is a disease in which the body has an inability to perform blood clot. Alright, so the protein which is supposedly used for blood clotting, alright, the normal protein which is used as a normal protein for blood clotting is somehow compromised. Okay, it is somehow compromised via inversion, and what happens to this protein is it affects the body, and the body now has an inability to perform blood clot. So in short, if a person has a bleeding, alright, if a person encounters bleeding here, it will not heal. Okay, it will not heal. Okay, it will not heal properly, just like a normal person's blood clotting ability. Alright, so it will not heal, and therefore the person will die bleeding. Okay, die out of bleeding. Okay, so numbers 1 has a inversion, and numbers 3 and 5 has an inversion. So as you can see here, if you will compare this particular lanes that you have on this picture compared to a normal, it is completely different. Okay, it tells us that the DNA segments that you have on these lanes are completely inversed. Okay, if you will compare them to 2 and 6 that you have over here. So the question here is, how does it happen? So it is something like this. Okay, so the segments of the DNA breaks and it is reinserted backwards. Okay, it is reinserted backwards. Okay, so in this case, we have here an example called hemophilia. Again, hemophilia is the inability of the body to perform normal blood clotting. Okay, because of a certain protein that was compromised and affects this whole process that we have over here. Alright, so that is inversion. Okay, so let's go now to the next example of gene mutation that we have right here. Alright, so we have here the translocation. So translocation occurs when part of the chromosome breaks off and it is added to different chromosome. So remember that chromosomes are made up of DNA. Alright, so made up of DNA, and this DNA contains a lot of segments. Now, if these segments break off, okay, if this segment break off and it is added to another segment, then definitely it will cause certain problems. Okay, so it will cause certain problems, and the problem always is the translation of amino acid. Okay, when you have a different translation of amino acid, definitely it will affect how the protein will function. So in this case, we have here the translocation. So how it is done? Okay, so translocation happens when a certain part of your chromosome, like the one that you have right here, was translocated into different place in that chromosome. Okay, so like the one that you have right here, Okay, so take a look at chromosome number 9. Okay, take a closer look at chromosome number 9 and chromosome number 22. So it was colorized so that we can identify the part of the chromosome that translocated. Okay, so in this case, we have the chromosome number 9. The part of the chromosome number 9 was translocated to chromosome number 22. So as you can see here, the part of chromosome number 9 is there, and the part of chromosome number 22 is on number 9. Okay? Now, in this case, there will be a serious effect. Alright? There will be a serious effect if this translocation happens. Okay? So, now, the first effect is, or the first common or the first rare disease that is caused by translocation of the part of the chromosome is the Alagille's syndrome. All right, so the Alagille syndrome is a syndrome that affects the heart, liver, and other parts of the body like the bones, okay, muscles, something like that. And most of the time, persons or individuals that has this kind of syndrome they have liver damage. Okay? Most of them have liver damage. Alright? Why? It's because this liver or the livers of this particular individual that has Alagille syndrome has an abnormality in the secretion of bile. Alright? There is an abnormal secretion of bile in their liver. So that is why it affects the functionality, the overall functionality of this organ. Okay, and eventually, if you have abnormal function of the liver, the ability to digest fats will be also affected. Okay, so the ability to digest fat will be affected, and also the functionality of your small intestine and your gallbladder will also be affected. Okay, so people with Alagille syndrome, all right, okay, most of them have a liver disease. Okay, so that's the first disease caused by translocation. So the second one is the Burkitt lymphoma. Alright, so the Burkitt lymphoma, so from the word itself, lymphoma, which means it affects the lymph nodes. And most people or most individual or some individuals that has this kind of disease, this is a rare disease. Okay, now most or some individuals that has this kind of disease has a impaired immunity. Okay, most common characteristic of this disease common characteristic of this disease is impaired immunity. Okay, once you have an impaired immunity, it can be fatal in the end. Okay, so this particular disease caused by translocation, like the one that we have right here, which is called the Burkitt lymphoma, yan. Most of them leads to cancer in the lymph nodes, and it causes impaired immunity. Okay, so Burkitt lymphoma. Alright, so the next form of gene mutation, the next example of gene mutation that we have right here is called the missense mutation. So what is a missense mutation? Missense mutation is a result of substitution of one amino acid, alright, or something like the change in the DNA base pair. Okay, there's a change in a DNA base pair that results in the substitution of a certain amino acid, okay, for another protein that is made by a particular gene segment. Okay, again, if there is a substitution, or if there is a change in the amino acid, if there's a change in the translation of the amino acid, then definitely the protein that will be produced here is malfunctioned. Okay, or there is a malfunction in the protein. Alright, so it looks like this. So again, this is the original base sequence that you have right here. And what happens here is the A, which is CAT, was replaced by C. Okay, it is replaced by C that you have right here. it is replaced by C. So instead of CAT, it becomes now CCT. Alright, CCT. So instead of having histidine, it is now producing an amino acid called proline. Alright, so now if histidine is the one needed, if histidine is the normal amino acid needed in this sequence, then therefore the protein that will be produced is somehow affected. Okay, so again, this is the missense mutation. Okay, so why is it called missense? Because there is no reason. Alright, so there is no reason why you can why you will change the sequence that you have right here. Okay, so there's no reason that you need to change the sequence that you have right here. So in this case, when you change that particular sequence, so logically speaking, it will affect also the amino acid that will be produced. Okay. Okay, example of missense mutation is the sickle cell anemia. Sickle cell anemia is a disease in which the body produces blood. Okay, so the body produces a blood cell that has an abnormal structure of hemoglobin. Okay, it has an abnormal structure of hemoglobin, then therefore the ability to store oxygen. Okay, just like a basic concept, just like all the basic concept that we have, that hemoglobin stores this oxygen which is needed by the body. So if you have a sickle cell anemia, the tendency is the ability of your body to store oxygen will be also affected, because the hemoglobin that you have on your blood is affected. Okay, the structure of this hemoglobin on your blood is affected by this kind of mutation. So therefore, the oxygen that was stored in this blood that you have right here, the one that you have a sickle cell over here, will be somehow different from the normal version of the blood cell that you have over here. Okay, so in some cases it is likely lesser than the normal. Okay, the oxygen that was stored, or the oxygen that will be stored in this type of blood cell that you have right here, will be lesser than the normal ones. Okay, so another example here is Tay-Sachs disease. Okay, Tay-Sachs disease. Alright, so Tay-Sachs disease is an example of a missense mutation in which it renders the body or it renders the ability of the body to digest or break down fatty substances. Okay, it renders the ability of the body to break down these fatty substances. There is an enzyme. Okay, there is an enzyme missing. Okay, in the process. So what will happen here is, if this one occurs in the brain, alright, if this one occurs in the brain, so it will affect the intellectual capability of this person or the individual, and also it will affect the growth and development of that particular organ, which is the brain. Okay, so some of the people that has this kind of rare disease that we have right here has an intellectual disability, something like that. Okay, and it has the brain. Okay, the brain of these individuals do not function the way it is normally doing. Okay, so that is the Tay-Sachs disease. Alright, so the next example of gene mutation that we have right here is the nonsense mutation. So in the nonsense mutation, one DNA base pair, okay, instead of substituting one, there's a changes in one DNA base pair, and what happens here is instead of substituting the amino acid, just like what we saw in the previous mutations, okay, it happens to be the base pair that was changed triggers or signals the cell to stop building the protein. Okay, you heard it right. It stops building this particular protein. So it looks like this. Alright, So it looks like this. So we have here a set of glutamines that is produced normally. Okay? Following the codons that you have on top. Okay? The codons of the DNA that you have on this segment. So glutamines all throughout. Now, in a nonsense mutation, what happens here is there is a change in the base pair, or there is a change in the codon or the nucleotide in the codon, and instead of having C, just like what you have on the original, it changes to become T. So it happens, or what will happen right here, it will change to U. Okay, if you transcribe this one into mRNA, okay, it will become UAG. Okay, it will become UAG in the process. So UAG is, if you look at UAG on the codon chart, it is translated as a stop. Okay, it will be translated as stop codon. Okay, so instead of creating more glutamines, the process will halt. Okay, so the process will halt, and then what happens here is the protein that will be created is malfunction because it was shortened. Okay, instead of having a long series of amino acid, it was shortened prematurely. So that is the nonsense mutation. All right, so the next example of mutation that we have right here is the frame shift mutation. Frame shift mutation is a type of mutation occurs when the addition or loss of the DNA base, okay, the addition or loss of base of the DNA changes how you read the frame. That is why it is called frame shift. So from the word itself, shift means change, all right? And the frame that you have right here, this is the DNA. All right, the DNA segment. Okay, so when you remove a nucleotide, it will affect how the gene is being read, okay, in the first place. Okay, so let's take a look at that. So the frame shift mutation that you have right here. So example, we have here the series of amino acid: histidine, serine, histidine, valine, leucine, methionine, then we have here the leucine. Okay, so this is the normal. Okay, so this is the normal group of amino acid, normal set of amino acid. So in this case, all right, so in this case that we have right here on the bottom part, there is something that was added, all right, or in this case it is not added. All right, it was not added. What happens here is, all right, so what happened here is the C was not read. All right, so the C that you have right here compared to the C on the normal version, the C on the normal version was read properly, and the C on the bottom version of this sequence was not read properly. So it happens to be it was skipped. All right, it was skipped, and what happens here is there is a shift in the frame that you have right here. Instead of having CAT, it will now have ATT. We have now CAC, we have now ACG, we have now TAC, we have now TCA, we have now TGC, and we have now TAT. Okay, in the process. So which is completely different from the one on top. Okay, so what do you expect? It will change the amino acid sequences. Okay, instead of having the normal, we have now the brand new. All right, we have now the brand new series or brand new sequence that we have over here of amino acid: isoleucine, histidine, rheonine, tyrosine, serine, cysteine, and tyrosine. All right, so if this one is not supposedly the normal, all right, if this one is not supposedly the normal version or the normally needed, so therefore this particular protein is malfunctioned. Okay, there is a malfunction on the certain protein that will be created on this kind of sequence that you have right here. So that is the frame shift mutation. Okay, so notice that if you have skipped one letter if you have skipped one letter, only one letter, it will affect the whole frame that you will read in this sequence. Okay, so examples of frame shift, or there are some examples of frame shift mutation. Okay, so the next example that we have right here is the repeated expansion mutation. Okay, so the repeated expansion mutation is a type of mutation in which the nucleotides are somehow repeated in several ways. Okay, so the nucleotides, instead of going into a normal sequence, it is repeated several times. Okay, just like what we have right here. Okay, again, this is the normal version of the sequence. Alright, so this is the normal version of the sequence, and what happens here is there is a portion of the sequence that was repeated. In this case, we have here the codon CAG. Okay, the codon CAG was repeated several times, so therefore, instead of having valine and isoleucine in the normal sequence, it becomes a series of glutamines. It becomes now a series of glutamines. So if you have a protein, okay, if you have a protein that was created in this kind of sequence that you have, then therefore this protein is said to be malfunction. Okay, the functionality of this protein will be affected if you have certain changes in the amino acid that was translated in the first place. Okay, so this is the repeated expansion mutation. Alright, next one, or the last one that we have right here in the gene mutation, is called the duplication. So from the word itself, duplicate, which means that the certain piece of the DNA was duplicated several times, and as we all know, if you have duplicated a normal sequence, then therefore the results will be the protein will tend to cause irregular function. Alright, it can have an irregular function. So just like what you see here, there's a duplication in a certain part of the chromosome, like the one that you have right here. There's a duplication. So what happened here is the chromosome on the other side. Alright, so the chromosome, the new chromosome that you have right here, is totally different in terms of length, and the length itself is different from the previous version. Okay, so what will happen if this section is duplicated? So therefore, the protein that will be produced by that section is somehow compromised. Okay, so the protein that will be produced by that section is somehow compromised. So example of this kind of mutation is called the Charcot-Marie Tooth disease. Okay, so this kind of disease is very rare. Okay, this kind of disease is very rare because this is an inherited disease. Okay, so inherited, which means that you can this disease can be passed to the offspring via parents. Alright, so this is a neuromuscular disease. This is a neuromuscular disease. So when we talk about neuromuscular disease, it is a disease which affects the nerve endings found in the muscles. Okay, so the nerve endings of this individual that has this kind of disease have been compromised, and it doesn't function properly. So the ability to move these particular parts of the body is somehow compromised. Okay, because the nerve endings, the muscles, which is supposedly functioning or normally functioning, is somehow affected by this duplication that happened in the chromosome. Okay, so this duplication, so the duplication created this particular or caused this disease that you have right here, the Charcot-Marie Tooth disease that you have right here. Okay, so that was the gene mutation and the examples that we have right here, which is the different examples that are under the gene mutations. Okay, so Up next is the chromosomal mutation. Alright, so what is the chromosomal mutation? Chromosomal mutation is a type of mutation which is said to be unpredictable. Okay, this is said to be unpredictable, and the changes brought about by the chromosomal mutations is because there are problems. Alright, there are problems that happened along the way. Okay, especially during meiosis. Okay, especially during meiosis. As we all know, meiosis is a type of cell division that occurs in the gametes. Okay, so this is the type of mutation that occurs in the gametes, like egg cell and then sperm cell. Now, the meiosis was the meiosis is affected, then therefore the number of chromosomes that was said to be normal is also affected. Okay, most of this chromosomal mutation is brought about by mutagens. All right, so it is brought about by mutagens as well. So like chemicals, radiation, something like that. Okay, it can definitely affect how the chromosome will look like. Okay, some chromosomal mutation can result to changes in the number of chromosome. Okay, in humans we have 46 number of chromosome. Okay, in humans we have 46 chromosome. It happens to be that if there is a chromosomal mutation, there will likely to have an extra chromosome. Instead of having 46, it might likely to have an extra, or it might likely to have a deficient number of chromosome. Okay, so missing our deficient number. Instead of 46, we have 45. All right, so this is chromosomal mutation. Okay, compared to the gene mutation, the gene mutation is on the segment of the DNA, and it affects how the amino acid will be produced. In the chromosomal mutation, it affects the structure, and somehow the the total number of chromosome is affected in this type of mutation. Alright, so under the chromosomal mutation, we have the autosomal aneuploidy. Alright, so which means the word aneuploidy means extra number. Okay, extra number of chromosome. Okay, so there is an extra number, or somehow there is a abnormality in the number of the chromosome. That is what the aneuploidy is all about. Okay, so this is the condition of having abnormal number of chromosome. It can be monosomy, alright, or it can be trisomy. Okay, so later we will try to find out how is this autosomal aneuploidy happens. Okay, so alright, so one of the most common reasons of autosomal aneuploidy. So since we are talking about autosomal aneuploidy, so what kind of cell division where autosomal aneuploidy is possible to happen? So it is in the meiosis. Only in the meiosis which you can separate. Alright, there is a separation between the chromosome, and the number of chromosome will be affected, so you will likely to have you will likely to divide the chromosome there in meiosis. So there is a particular reason why the autosomal aneuploidy happens, and that is the errors in disjunction. Disjunction happens, alright, so disjunction happens when the chromosome failed to separate properly during meiosis. It only happens during meiosis. Alright? So it only happens during meiosis, this disjunction process. Okay, now what are these homologous chromosomes? Homologous chromosomes are chromosomes of the same structure, same length, same type, okay, and same location. Alright? These are homologous chromosome. Now, when the homologous chromosome failed to separate properly, that is now called as non-disjunction. Alright? Again, that is called non-disjunction. So how does this non-disjunction happens? Alright, there are two scenarios here. Alright? So the first one, we have a normal set of chromosome over here. Alright? So supposedly we are pertaining to humans. Okay, so we have 46. Okay. Now, the first scenario that you have right here is that there's a normal separation of the chromosome. So, there's a normal disjunction here. Okay? So, there's a normal disjunction. So, the two chromosomes, the two homologous chromosomes separated. But, the non-disjunction happened on the second meiotic division. Okay? So, the second meiotic division, this is where the non-disjunction happens. So, it happens to be that, supposedly, the sister chromatids will tend to separate on the second meiotic division. It happens that it didn't separate. So, there is a non-disjunction. So, what happened here is, this cell is incomplete. Alright? So, this cell that you have right here is incomplete. While the other one has an extra number of chromosome. Alright? It has an extra number of chromosome. This one is incomplete, and this one has an extra number of chromosome. So, should there be an effect? Yes, there is an effect on that. So, I will show you later what are some examples of different effects of non-disjunction. Okay, in terms of number of chromosomes. Okay, so the next scenario that we have right here, the second scenario that you have, is on the first meiotic division. Alright, on the first meiotic division, the chromosome or the homologous chromosome didn't separate properly. Alright, so it didn't separate properly, so what do you expect? What do you expect to happen? So, this one contains an extra number of chromosome, and this one contains a missing amount of chromosome. Okay? So, it is incomplete. Okay? The number of chromosomes that you have on this cell is incomplete. Okay? So, will there be an effect on the second meiotic division? Will there be an effect on the second meiotic division? Yes, definitely there is an effect. Why? It's because since you have an extra number of chromosome that you have right here, since you have an extra number of chromosome, it will be, it has a profound effect on the second meiotic division. So what you can see here, you have a lot of chromosome that has an extra number on it. Okay? You have a lot of cells that has an extra number of chromosome on it. So in this case, we have now 24. Which should be 23. Okay? Which should be 23 only. Alright? So, in the other group of cells, like the one that you have right here, since the cells from the first meiotic division doesn't have a complete number of chromosome, so therefore, the cells in the second meiotic division will also be affected. In the process. So, the cells in the second meiotic division that also have an incomplete number of chromosome. Okay? So, in this case, we have 22. So, if this is fertilized, okay? If this is fertilized by a sperm cell, if this is fertilized by a normal sperm cell, the count or the normal count of chromosome will be affected. So, what does the offspring look like? Okay? So what does the offspring look like if you have an extra number of chromosome or an incomplete number of chromosome? So the first case that we have right here is the case of an extra chromosome in number 21. Okay? So the number 21 that you have right here, the case that you have in this picture. So instead of having 2, we have 3 chromosomes on number 21. So that is why it is called trisomy 21. Okay? So it is also known as Down syndrome. Okay? It is also known as Down syndrome. So these are the notable characteristic of the person that has a Down syndrome. Okay? So flattened nose, flattened nose and face, the upward slanting of the eye, something like that. In terms of the appendages, so the finger is curved inwards, and on the foot, so we have here increased skin creases, okay, on the foot that you have right here, okay, so on and then so forth. All right, so Okay, Down syndrome is affected by the mother's age. Okay? As the mother age increases, the incidence of having Down syndrome per birth also increases. Okay? So, okay, that's how the mother's age should be taken for should be accounted for here. Okay? So, as you can see here, the person that has, or an individual that has a trisomy number 21, okay, is completely different in terms of facial features compared to a normal person. Okay? So, these people are sterile. Alright? So, these individual are sterile. They are not capable of producing an offspring. Okay? So, that is the Down syndrome. Alright, another case that we have right here is the trisomy 18. Alright, so from again from the word trisomy means an extra chromosome on number 18. Okay, so in this case we have the number 18 chromosome over here. Okay, so instead of having 2, it has 3. Alright, it has 3 chromosome. Alright, so what do we have right there? Okay, so the disease that it causes is called the Patau's syndrome, or Edwards. Alright, rather Edwards syndrome. Okay, so Edwards syndrome. So this is the trisomy number 18, and these are the sets of characteristic that a person that has an Edwards syndrome, okay, should you should take note of. Okay, so it has small head like the one that you have right here. So, an occurrence of cleft palate, okay, on the offspring. Okay, underdeveloped thumbs and nails, undescended testicles. So, since they have undescended testicles, okay, in males, so therefore, people or individual that has this kind of syndrome, which is called the Edwards syndrome, has a is or are sterile. Again, they are sterile. Again, sterile means inability to produce an offspring. Okay? So, that is trisomy 18. Another case that we have right here is the trisomy 13. Okay? So, the trisomy 13, instead of having 2, we have extra chromosome on the 13th chromosome that you have on this picture. So, what kind of disease it causes? Alright, so this is the Patau's syndrome. Okay? So, this is the Patau's syndrome. So, I'll write it right here. Okay, so Patau syndrome, so most of the people or most of the individual that has a Patau syndrome is born prematurely. Okay, so they are born prematurely. Now, if they survive, all right, if they survive, you will notice some characteristics that you have right here. Okay, you will notice some characteristics that you have over here, and individuals that has this kind of syndrome is again sterile. Okay. So, if they survive, all right, if they survive after being born prematurely, okay, so you see on the picture, all right, you see on the picture that this individual that has this kind of extra chromosome on number 13 has a microcephaly, so small head, and we have a low set of ears that you have over here, and somehow the fingers and the appendages have a overlapping features. Okay, and aside from that is there are some notable heart defects. Okay, and the brain development is also affected by this disease. Okay, so it is very hard from an individual to survive this kind of disease that we have right here. So this is the trisomy number 13, or also known as the Patau syndrome. Alright, so if you have trisomy, we have monosomy. Okay, so monosomy means deficient number of chromosome. Okay, so missing number of chromosome. So in this case, we have Turner syndrome. Okay, so the Turner syndrome. So as you can see here, the Turner syndrome, the one that is affected on the Turner syndrome is the sex chromosome over here. Okay, so the sex chromosome over here is affected. So instead of having X and Y, it is X and 0. Alright, X and O. Okay? So, in this case, we cannot identify if if this person is a male or a female. But definitely, all people are born when we are on our mother's womb. Okay, an individual is born female. All right. Later on, it will become a testicle if the individual has a Y chromosome. So definitely, this person is a female. But it is not guaranteed that the female reproductive organs of this person is completely developed. So that is the Turner syndrome. Okay, so let's take a look on what the Turner syndrome looks like. Okay, so this is a person. So this is a artist rendition of the person who has a Turner syndrome. Okay, so rudimentary ovary. So again, as I've said earlier, it has a undeveloped. It has a an undeveloped structures. Okay, so especially the female reproductive structure that you have right here. So poorly developed breast. So we have constricted aorta. Okay, on the heart, small fingernails over here. All right, brown spots over there. Okay, so there are some. These are some some unusual facial features that we have over here. So this is what you should look for if a person has a Turner syndrome. Okay, again, they are sterile. Okay, these people or this individual that has Turner syndrome is sterile. All right, so next next that we have right here is the Klinefelter syndrome. Okay, on the Klinefelter syndrome, the normal sex chromosome, this is a trisomy. Alright, so this is a trisomy. This is not a monosomy case. This is a trisomy case. In this case, it doesn't happen on the number 1 to 22nd chromosome. It didn't happen right there. The trisomy, okay, so the extra chromosome is seen on the sex chromosome over here. Instead of having XX or XY, all right, instead of having XX or XY, it happened to be that it has two X's. All right, it has two X's and a Y chromosome. So therefore, it has 3 sex chromosomes. So, what is this? What is the characteristic of the person who has a Klinefelter syndrome? Okay, again, this is a product of non-disjunction. Okay, this is a product of non-disjunction, and common characteristic of the person who has Klinefelter syndrome is longer fingers and arms. It has a unusual skin features, and it has intellectual challenges, all right, or intellectual disabilities. And most of the people that has this kind of syndrome has a normal lifespan. Since it is only the sex chromosome that was affected, okay, it is only the sex chromosome that was affected, they tend to have normal lifespan compared to those people that has trisomy 13, trisomy 18, and also the Turner syndrome. So, okay, so these people tend to live a normal lifespan, and most of them has undeveloped sex organs. Okay, in this case, if you have extra X, alright, if you have extra X over here, what happened here is you are a female. Okay, you are a female but you have an underdeveloped male organs. Okay, so therefore you are sterile. Okay, so therefore an individual who has this kind of syndrome is sterile. So they do not have a ability to produce an offspring. Okay, that is the Klinefelter syndrome. All right. So another kind of mutation that we have right here, which is autosomal recessive, which means that this kind of mutations can be hereditary. All right. It can be hereditary and it can be passed through certain generations. Okay, via the offspring. Okay. So in this case, we have here the albinism. Albinism is a type of mutation in which the person has an inability to produce melanin. Alright? On the skin, the hair, and even the eyes. Alright? So, these people are very vulnerable to skin cancers when they are exposed to ultraviolet light. Okay? So, albinism. Okay? Okay, so there are a lot of autosomal. Okay, there are a lot of autosomal disorders, okay, that can be associated in our chromosome that we have right here. I cannot discuss them all. I will just show them all to you here in this picture. So we have here the ALS, so the nerve degenerative disease that we have, and it happens on the 21st chromosome. If there is a mutation, alright, if there is a mutation that was passed over the generation on this chromosome, then therefore the offspring might likely to have an ALS. Okay? So one of the most familiar that we have right here is the breast cancer. 5% of the cases is associated to the 17th chromosome that you have right here. Okay, most of the breast cancer are hereditary. Okay, it can be transferred or you can inherit that particular disease from your parents. Alright, another one here is the polycystic kidney disease. Alright, so this is a type of disease on the kidney in which the kidney grows, or there are some cysts. Okay, there are some tumors that grows abnormally on the kidney. Okay, so it is associated to somehow chromosome number 16. Okay, so mutations in the chromosome number 16 might likely to cause this kind of disease. Okay, another one is Alzheimer's. Okay, Alzheimer's. So the Alzheimer's disease is a degenerative brain disorder, and most of the time, according to some research, most of them occurs in the chromosome number 14. Okay? So if there are mutations in the chromosome number 14, expect that Alzheimer's disease can be observed on those individuals. Alright, so we have here the sickle cell anemia. Alright, so as I've discussed earlier. Okay, so next one we have phenylketonuria. Okay, so this is the inability of the body to recognize the phenylalanine. Alright, which is an amino acid, an essential amino acid, which is needed in order for your brain to develop properly. Okay, so that is the phenylalanine. So if your body doesn't recognize that kind of amino acid, then therefore there will be an intellectual disability that will happen on that individual, or mental retardation. Okay. Alright, so we have the melanoma. It is associated to chromosome number 9. Cystic fibrosis on chromosome number 7, and many more. Alright, so that is, alright, so these are the mutations that happen on the human chromosome. Okay, so certain mutation will cause certain disease on the human chromosomes. Okay, that we have over here. Okay. Now, other forms of genetic disorders that we have right here. Okay, so these are somehow inherited. Okay, these are somehow inherited, and some of them are caused by external factors. Okay, for example, the cleft palate that you have right here. Alright, so the cleft palate and the diabetes that you have right here. Okay, so cleft palate is the inability or underdevelopment of the skull region. Okay, this is the last region. The middle of your face is the last region that will join in the fetal development. Okay, so if you have cleft palate, so meaning to say that the skull region, the middle part, alright, of your skull, failed to connect properly. Okay, failed to connect properly, so therefore, individuals develop a cleft palate at the end. Okay, so you have some underdeveloped lip on those people. Alright, another one is diabetes. Alright, diabetes can be caused by hereditary or external factors. Okay, so if you eat a lot of carbohydrates or like sugar, so you might likely to have a type 1 diabetes. Okay, there are some people that has a hereditary form of diabetes. Okay, so again, it is some form of a mutation in the cells in the pancreas. Okay, another one, the heart disease. Okay, so heart disease can be inherited, and some of them can be caused external. Okay? So, heart disease, or if there are some mutations on your cells that are found in the heart, then therefore the functionality, the overall function of your heart will be also affected. So infertility also is a genetic disorder. Okay? So obesity. All right? So there are some diseases which is caused by a mutation in a certain chromosome that enables your body to automatically produce cholesterol. Okay? There are some diseases like that. So your body automatically develops this cholesterol on your bloodstream. Okay? And it can cause obesity. Alright? So something like that. Okay, intellectual disability, something like that. Cancers. Okay? Very familiar form of mutation. Cancer. Cancers can be caused or can be inherited. And sometimes it can be caused by external factors like radiation, carcinogens, or even infectious agent like viruses or bacteria. Okay? So, these are some forms of genetic disorders. I cannot discuss them all here on this video. So, there are a lot of resources that are present in the internet that can show you these disorders that we have right here. Now, as a final conclusion on this lesson that we have right here, mutations can neither be good or bad. In this case, in this discussion that we have right here, mutations that are caused by ionizing radiation, that are caused by carcinogens, or even caused by viruses, sometimes they end up bad effects. Okay? So this mutation causes bad effects, like the one that you have right here, the genetic disorders that we have discussed. Okay, those are the bad effects. But there are times that mutation causing good effects. Okay, like the one that we have are, if you are a bacteria, the good effects of mutation is antibiotic resistance. Okay. Okay, nowadays we have the COVID-19, so the COVID-19 continues to mutate. Okay? So if you are a COVID-19, that is a good mutation on you. Alright? So other mutations like sickle cell anemia is a good mutation in the sense that it protects the person from malaria. Okay? So this malaria, okay, so this disease doesn't contract or doesn't infect people who has sickle cell anemia. All right. So there are some resistances that will be developed. Okay. So those are some good effects. And mutations, okay? So on the next video lesson that we will have in the future, mutations are the agents of evolution. Okay? Without mutation, organism will not change. Okay? Organism will not change, and therefore they will not evolve. Okay, and they will not be able to adapt to a certain environment that they will live in. All right. So again, mutation is neither good or bad, and sometimes mutation is neutral. Okay. So that's it for this mutation lesson that we have over here. And that ends up our topic for this video lesson for today. I hope you'll be able to get value on the topic we discussed. And if so, don't forget to like this video. If you have questions, suggestions, write them down in the comment section below this video. It will help me a lot in the algorithm for my YouTube channel. Most importantly, don't forget to subscribe to my channel and hit the notification bell for you to be updated for more science video like this in the future. I'll see you next time, everyone. Enjoy learning, stay safe, and peace out.